Welcome back. We're decoding the health matrix.
A simple solvent can cross biological barriers, help preserve living cells during freezing, and change how other substances move through tissue. So why is dimethyl sulfoxide, or DMSO, still unfamiliar to most people? And why, when it does come up, does the conversation move so quickly between forgotten medical breakthrough and dangerous cure-all? The answer is more useful than either extreme. DMSO has unusual chemistry. Some of its medical uses are already established. Its interactions with membranes, inflammation, and tissue transport open doors that researchers and clinicians are still walking through. And the molecule's real story is more interesting than the extremes allow. Today, we will make DMSO easier to understand. We will look at where it already has legitimate medical value, where its anti-inflammatory and tissue transport potential is pointing next, and why the best results come from careful formulation, route, and clinical context. DMSO stands for dimethyl sulfoxide. Its chemical formula is C2H6OS. At room temperature, it is a clear liquid with a distinctive sulfur-like odor. It is also a powerful solvent, which means it can dissolve or interact with a wide range of substances. The compound was identified in the 19th century by the Russian chemist Alexander Zaitsev. Decades later, the pulp and paper industry encountered DMSO as a byproduct of processing wood. Its ability to dissolve materials and move through them made it useful in industrial settings. That industrial history led to an important medical question. If DMSO can move through materials so effectively, what does it do in living tissue? In the early 1960s, surgeon Stanley Jacob and industrial chemist Robert Hirschler helped investigate its biological properties. DMSO was studied in connection with pain, inflammation, wound healing, tissue injury, and cell preservation. In 1964, Life magazine called it a new wonder drug. The excitement was understandable, but the evidence was still developing. Animal studies raised concerns about changes in the eye lens at 50 to 100 times normal dose exposure, and those changes were reversible. In 1965, the United States Food and Drug Administration suspended clinical investigations while safety questions were examined. Later, the FDA approved a prescription DMSO product, RIMSO 50, for a specific use, intravesical treatment of interstitial cystitis, a painful bladder condition. That approval matters. It shows that DMSO has a legitimate clinical role. It also marks a starting point, not a ceiling. One approved indication is proof of medical value. It is also an invitation to keep asking what else this chemistry can do under the right conditions. The most useful way to understand DMSO is as a molecular ferry. A ferry can carry passengers across water. DMSO can interact with both water-loving and lipid-loving environments. That allows it to move between watery tissue spaces and the fatty layers of cell membranes. In some settings, it can help other molecules travel through tissue. This membrane interaction is one reason DMSO remains medically exciting. It can influence how membranes behave and how substances move across them. In topical research, that property has made DMSO a vehicle for other compounds, opening paths that ordinary solvents struggle to reach. In cellular preservation, its interaction with water and membranes helps protect cells during freezing. But a ferry does not decide whether its passengers are helpful. It simply carries them. DMSO may carry unintended medication. It may also carry unwanted substances, contaminants, dyes, residues, or irritants. That single property explains both its medical potential and the care required around it. Used carelessly, it can carry the wrong cargo. What is reasonably established? DMSO can penetrate skin and enter the bloodstream. It interacts with cell membranes and can alter membrane behavior. Exposure can produce a garlic-like or onion-like taste and smell because of the compound and its metabolites. These effects are properties of the molecule. DMSO can also interact with containers and other materials. Compatibility depends on the particular formulation, concentration, temperature, contact time, and material. Some plastics may soften, swell, or release compounds after exposure to solvents. That is why formulation is part of the medicine. Purity, sterility, storage, concentration, container compatibility, and route of exposure all matter. DMSO from an industrial setting is not automatically suitable for medical use. A label that says pure or pharmaceutical grade does not replace regulated guidance, verified quality, or a clear clinical indication. The ferry analogy is practical here. If the ferry is contaminated, or if it picks up material from an unsuitable container, the passengers may not be what anyone intended. The FDA-approved use in the United States is intravesical DMSO for interstitial cystitis. In that setting, a healthcare professional uses a regulated pharmaceutical product inside the bladder. Clinical studies and later real-world data have examined symptom relief, tolerability, and treatment response in selected patients. Beyond that labeled use, clinicians and researchers have explored DMSO in other settings. Topical preparations for musculoskeletal pain, as a vehicle that helps other medicines reach tissue, and in experimental work on inflammation and injury. Off-label simply means an approved product is being used in a way that is not the specific indication on its label. It does not mean the idea is empty. It means the evidence, formulation, risks, and patient context still have to be weighed by someone qualified to do that weighing. DMSO has another major and well-established role in cryopreservation. When living cells freeze, ice crystals and changes in water concentration can damage membranes and internal structures. DMSO can reduce some of that damage by entering cells and changing how water behaves during freezing. It has been used in the preservation of hematopoietic stem cells, immune cells, cultured cell lines, and other biological materials. It also appears in research and cellular therapy workflows involving specialized cell products. This is one of the quiet triumphs of the molecule. Under carefully controlled conditions, DMSO helps living tissue survive a process that would otherwise destroy it. Cells are frozen according to defined protocols, then processed and administered through specialized medical or laboratory systems. That setting does not make DMSO a general health supplement. It does show something powerful. When the process is controlled and the goal is clear, DMSO can protect life at the cellular level. The value comes from the chemistry, the controlled process, and the specific biological goal. Now to inflammation. DMSO has been studied in relation to swelling, inflammatory signaling, nerve excitability, and tissue injury. Laboratory and animal research suggests that it may influence pathways involving reactive oxygen species, prostaglandins, cytokines, and histamine. Its redox chemistry is part of the interest. DMSO can participate in oxidation and reduction reactions. Its metabolites include dimethylsulfone, commonly known as MSM, and smaller amounts of dimethyl sulfide. These findings give DMSO biologically plausible anti-inflammatory potential. They help explain why researchers have kept returning to it in tissue and pain models, and why clinicians who work with inflammatory conditions continue to take the molecule seriously. Inflammation is not a single switch. The effect of a molecule depends on the tissue, the exposure, the timing, the route, and the other substances present. What makes DMSO compelling is that several of those levers appear to move in a useful direction at once, and it is why the anti-inflammatory story keeps earning new chapters. So, the evidence should be described in layers. The chemistry is established, cellular and animal effects are documented and biologically coherent, some clinical applications already exist, broader uses are still being mapped, and that mapping is part of what makes the field feel alive. Pain is one area where DMSO has attracted sustained attention, and for good reason. Topical preparations containing DMSO have been studied for musculoskeletal pain, including osteoarthritis. Some research has examined DMSO as a vehicle for topical anti-inflammatory medicines. That distinction matters, and it is also part of the opportunity. Any observed benefit may come from the active medicine, the vehicle, their combination, or the treatment context. DMSO's ability to help other compounds reach tissue is itself a therapeutic asset. Historical reports and experimental studies have also involved headaches, tendon pain, nerve pain, and other conditions. Those observations keep the door open. They invite careful clinical judgment and better designed studies, not dismissal. The responsible claim is both modest and hopeful. DMSO has plausible effects on tissue and nerve biology. Some applications already have clinical evidence, and the condition-specific map is still being drawn. Trauma and neurological disease sit at the edge of what is known, and that edge is worth watching. DMSO has been discussed in relation to brain and spinal injury because inflammation, swelling, blood flow, and oxidative stress are central to tissue damage. Animal and laboratory work has produced mechanisms that researchers find encouraging: reduced swelling, altered inflammatory signaling, and protection under oxidative stress. These findings have not made DMSO standard treatment for traumatic brain injury, spinal cord injury, stroke, multiple sclerosis, or other neurological emergencies. Urgent, evidence-based care remains the first obligation. At the same time, the mechanisms are not empty. They are reasons serious investigators keep DMSO on the research agenda for conditions where inflammation and membrane injury do real harm. The same discipline applies to claims involving complex diseases such as schizophrenia, Down syndrome, Alzheimer's disease, cirrhosis, cardiac disease, and other conditions sometimes listed in advocacy materials. A proposed mechanism or an old report is not enough to call something a treatment. It can be enough to justify curiosity, careful study, and an honest conversation about what the chemistry might one day support. Eye disease is another area where claims need careful sorting, and where the history is more nuanced than the headlines. Older animal studies raised lens concerns at high exposures. Those findings helped trigger the 1960s pause, and they deserve to be remembered accurately. The exposures were extreme, and the changes were reversible. More recent experimental work has explored DMSO in models of retinal disease, including a rat study involving subconjunctival injection. That is, a research finding in animals. It is not evidence that DMSO prevents cataracts or restores vision in people. It is a signal that the eye remains a place where DMSO's chemistry is still being tested with intention. Cancer research has examined DMSO as a solvent, a differentiation-inducing agent in some experimental systems, and a possible component of drug delivery or supportive care. These are legitimate research questions with real stakes. They do not mean DMSO treats cancer or replaces oncology care. They do mean the molecule keeps showing up wherever scientists need a way to move compounds, change cell behavior, or protect tissue under stress. A plausible mechanism can point toward a useful research program. In DMSO's case, those programs keep multiplying. The safety section deserves the same attention as the science, because safety is what makes long-term use possible. DMSO's ability to carry substances through skin is a double-edged property. If the skin, container, applicator, or mixture contains contaminants, DMSO may help move those contaminants inward. That is exactly why quality matters so much. The same ferry that delivers a useful medicine can deliver a problem if the cargo is wrong. Unknown mixtures and industrial-grade products are inappropriate for self-treatment. Do not assume that a supplement, essential oil, topical medication, or household chemical because DMSO carries it or because it has been diluted. People may experience skin irritation, redness, itching, burning, or other local reactions. The characteristic odor can persist and may be socially difficult. Taste changes or a sulfur-like flavor may occur after exposure. DMSO can also affect how other medicines move through tissue, creating the possibility of interactions. The history of lens findings in animals should not be exaggerated, but it should not be erased. High-exposure animal studies involved species, concentrations, and conditions that do not map neatly onto every human use, and the changes observed were reversible. The appropriate response is interpretation, proportion, and continued attention to safety. Anyone who is pregnant or breastfeeding, caring for a child, taking prescription medicines, managing kidney or liver disease, or dealing with a serious medical condition should discuss DMSO with a qualified clinician before exposure. Professional guidance is especially important for use near the eyes, on damaged skin, or inside the body. That conversation is not a break on possibility. It is how possibility becomes responsible practice. A related compound often enters the conversation, MSM, or methylsulfonyl-methane. MSM is chemically related to DMSO and is one of its metabolites, but it is a distinct compound with different properties. It is sold as a dietary supplement, often in products marketed for joint comfort. Research on MSM has produced mixed and condition-specific findings, and for some people, it has become part of a broader joint support routine. Its presence in the same chemical family does not make it interchangeable with DMSO. Evidence for one cannot simply be transferred to the other. Knowing the difference helps you evaluate each molecule on its own terms. The ferry analogy helps again. MSM is not the same ferry with a different label. It is a different molecule with different passengers, routes, and biological behavior. So where does that leave us? DMSO deserves serious medical attention because its chemistry is real. Its role in cryopreservation is important and ongoing. Its FDA-approved bladder application is specific and legitimate. Its membrane interactions and anti-inflammatory potential continue to open research paths that matter for pain, inflammation, and tissue injury. The key is to keep the claims in proportion without draining them of hope. A mechanism is not yet a treatment, but it can be the start of one. A laboratory result is not a clinical trial, but it can justify the next study. An animal finding is not a human recommendation, but it can light the way toward one. A long list of conditions is not the same thing as a body of reliable evidence, and building that body is exactly the work still worth doing. DMSO became difficult to evaluate because regulatory caution, uneven research, practical drawbacks, and ambitious claims became tangled together. Separate those strands, and the molecule itself becomes clearer and more compelling. It is a molecular ferry. It can carry valuable passengers. It can carry unwanted ones. Its usefulness depends on where it goes, what it carries, how it is formulated, and who is guiding the process. The responsible path is neither blind dismissal nor blind adoption. It is better research, responsible products, clear labeling, careful clinical judgment, and an informed conversation with a qualified clinician. Done that way, DMSO is not a closed chapter. It is a molecule still earning its place in modern medicine. That is where DMSO belongs, not in the category of miracle cures, and not in the category of forgotten curiosities, but in the more demanding and more promising space where real chemistry meets responsible medicine. and where the next useful application may still be ahead of us.
Thanks for listening. Keep decoding.